Hello everyone, my name is Krishna and today I will be presenting our research on characterizing the culturable bacterial communities from imported date fruits to control Listeria monocytogenes. So I'll start off by giving you a brief background of who I am. I am a MSc student in food science from the University of Guelph, and we do our work at CFREF, the Canadian Research Institute for Food Safety. And I am a student as part of Dr. Farber's group. This is him over here, who's also here with us today. And the University of Guelph is a Canadian institution near Toronto. And the work we do is part of the Food from Thought initiative and is funded by the Canada First Research Excellence Fund. In terms of the outline for the presentation, I'll be providing a brief background as to why we're working with these particular foods, which will then segue into our research rationale. I've then broken up the presentation into our 3 core objectives of the abstract. The first to explain why we're working with date fruits, the second to assess whether the bacteria from date fruits could impact the growth and/or inactivate L monocytogenes, and the last to assess the safety and novelty of the inhibitor strains we acquired from our second objective for applications in the food industry. I'll then wrap up the presentation by discussing future work and applications of this research. So the work we do uses food microbiology for the applications of food safety. And to give you a little perspective on food safety, as a statistic from the World Health Organization says that 1 in 10 people in the world fall ill after eating contaminated food on a yearly basis. And we're thinking a possible way to help combat this statistic would be to better understand the microbial community of ready-to-eat foods in the market. So with ready-to-eat foods, first off, they can support the growth of pathogens such as Listeria monocytogenes. And if we're looking at common antimicrobial control measures for these pathogens, we have the use of physical inactivation with either heat or pressure, the use of organic acid derivatives, and also biocontrol measures. And among these two, we'll be focusing more on bacteriocin proteins and the live bacteria that produce them, as we're speculating that the natural microbial community of ready-to-eat foods, they may also prevent pathogen growth, as they may also support the growth of these beneficial, such as anti-Listeria bacteria, and they may help us find novel bacteriocin proteins. Thus, we hypothesize that the microbiome of imported ready-to-eat date fruits will contain culturable bacterial members that possess antagonistic properties against the foodborne pathogen Listeria monocytogenes. We decided to look at imported fruits to increase the probability of finding these unique novel bacteria that haven't already previously been discussed for these applications. And in terms of L monocytogenes, it is considered to be one of the most severe foodborne pathogens, especially if you do become invasively infected with it, as it's known for having one of the highest case fatality rates. So on to our first objective, to essentially explain why we're looking specifically at date fruits. We initially surveyed a variety of different imported ready-to-eat foods. This includes dried apples from Argentina, cumin and fennel seeds from India, date fruits from China, Tunisia, and UAE, pistachios from USA, pollen from Poland, raisins from South Africa, and seaweed from China and Japan. What we did was we washed and massaged these foods to collect the bacteria both from the exterior and interior of these foods, and we then plated that wash on blood agar, and we collected isolates from this wash in triplicate per observable phenotype. After collecting all of these isolates, we then subjected them to a growth inhibition plate assay that we designed in-house. The way that this assay works is that we would first plate a lawn of an overnight culture of the pathogen, and then on top of that lawn, we would then drop an overnight culture of the different isolates we collected from our foods. We would allow both the isolates and the pathogen to grow together overnight, and the following day we would inspect the plate to look for these zones of inhibition. This is an area on the plate where the pathogen was not able to grow, and isolates that produce these zones, we would classify them as being an inhibitor strain, and those isolates that did not produce this zone, we would consider them to be a non-inhibitor strain. We are also able to optimize this assay to test for 16 different isolates at once by using a drop volume of 5 microliters from that isolate's overnight culture. This next table summarizes all this information from these foods. This column shows the total number of isolates we collected from these foods using our blood agar method. So we were able to collect 262 isolates. This next column shows a number of inhibitor strains as per the growth inhibition assay from the prior slide. So of those 262, 64 of those isolates were inhibiting the growth of L-monocytogenes. And this last column shows the identity of these inhibitor strains as per 16S rRNA sequencing. And if we're looking at the data from date fruits, we're seeing that we're able to collect a large number of isolates from these dates with our blood agar method, of which a good number of them are inhibiting the growth of L-monocytogenes, and these inhibitor strains are also coming from a broad diversity of genera. So we decided, let's further pursue date fruits for the remainder of this project, and that's what we did. So moving on to our second objective, to assess whether the cultural members from these date fruits can impact the growth and or inactivate L-monocytogenes. So what we did was we collected fresh batches of dates from retail stores, and we were able to find date fruits from 5 different imported regions. We acquired dates from China, Iran, Palestine, Saudi Arabia, and Tunisia. And we did the same thing. We collected isolates using our blood agar method, and we detected inhibitor strains using our growth inhibition plate assay. From doing so, we were able to collect 191 isolates, of which 35 of them were found to be inhibitor strains, which is quite a lot. That's close to 20% of our collected isolates are inhibiting the growth of L-monocytogenes. And after we sequenced all of these inhibitor strains, they were all found to identify as Bacillus species. Specifically, these Bacillus species were found to identify across 6 distinct groups of species. And this next figure shows us that data. It shows these 35 inhibitor strains organized across these 6 different species groups and plotted against the zones of inhibition that they produced. And a couple things you can note, the bulk of our inhibitor strains, they're coming from species groups 1, 2, and 4. And our best inhibitors, so those that produce the largest zones of inhibition, are coming from groups 1 and 2. Nonetheless, though, from these 35 inhibitor strains, they were found to produce inhibition zones that range from 0.5 to 5.7 millimeters. Moving on to our third objective, to assess the safety and novelty of these inhibitor strains for applications in the food industry. So let's go back to this figure. We found that all of the isolates from our first species groups are all associated with the gene for the pumalacidin toxin. It's an undesirable toxin for food industry and for human health. So we decided let's not pursue any of these isolates. We also found that 4 isolates from group 4, they identified with an undesirable species that could be potentially associated with human illness. We decided let's not pursue those 4 isolates either. We also found that the 2 isolates from group 5, they were associated with an extensively studied species. So we figured that they would lack novelty if we decided to further pursue them. So what we did then was, with our remaining isolates with our remaining species groups, we decided to look at the top inhibitor strain from those remaining groups to further pursue for virulence testing, whole genome sequencing, and antimicrobial activity. This leaves us with 4. I do have my virulence work completed, but we finished it after this abstract was submitted. But I'll briefly tell you how we're going about our model for virulence, and I'll touch upon briefly the results we acquired as well. So for our virulence work, we're assessing whether those selected inhibitor strains could also impact the virulence of this pathogen. And we're doing this using a fluorescence reporter assay, where we are subjecting the supernatant of these inhibitor strains as a treatment to different L-mono reporter strains. We have a wild type, a delta perf A as our negative control, and a perf A star as our positive control. And perf A is known as a master regulator for virulence of this pathogen. So it makes for an excellent biomarker to target when studying the virulence of this pathogen. And each of these strains, they contain a plasmid which has a gene for a red fluorescent protein, which is downstream of a promoter that is tightly controlled by perf A. Thus, you'd only get expression of fluorescence if you get expression of perf A. They're very tightly interconnected with one another. So if we're looking at our wild type, after being subjected to this treatment, if we're still seeing fluorescence, that's still telling us that our strain is still active in terms of virulence. However, if we're seeing a reduction in fluorescence or a complete elimination of fluorescence, that's telling us that there's something from this treatment that's impacting the virulence of the strain. And we found that for those 4 selected inhibitor strains, they were all found to significantly reduce the expression of PERF-A from the wild-type strain, which is very exciting to find for us. In terms of our future work, we're looking to conduct whole genome sequencing on these selected inhibitor strains, and we're also looking to characterize the anti-listeria compounds produced from those selected inhibitor strains using MALDI-TOF mass spectrometry. And we also wanted to do tolerance tests on these anti-listeria compounds against different temperature, enzyme, and pH conditions. In terms of applications of this work, this could lead to the discovery of novel antimicrobial metabolites or bacteriocins, or even beneficial bacillus species that could be added to foods to inactivate and or control this pathogen. I'd just like to take a moment to give a very warm thank you to all of the scientists at our Food Safety Institute. I also wanted to point out the other authors of this abstract, and I also wanted to point out 2 other students who are here with us today that are giving poster presenters. And I included their poster numbers in brackets. They have their posters here today. So if you do get the chance, I do highly suggest for you to check out their posters to learn more about the exciting work our group is doing. And with that, I thank you so much for listening to my presentation. Now I'll open the floor up to any questions you may have.
Thank you very much for the nice presentation. I guess that the red fluorescence protein reporter strain was the present for Christmas. Okay, so I open the floor to questions, please. Go ahead.
Hi, very nice presentation.
Thank you very much.
You're looking at the virulence of the effect of the virulence of bacteria. But you also check the effect of the bacteria itself on, let's say, human cells to see whether it's safe to.
That would be a good idea to further assess the safety of these inhibitor strains. That would be outside of the context of my project, but it would be a great idea to do maybe for like another project, for another student, if they were to continue on this work.
Okay, there was a question.
Do you think to test antimicrobial activity against other pathogens?
Again, yeah, this is just a master's project. We had to leave our so we just looked specifically at this pathogen. I want to finish on time.
Did you do a whole genome sequencing to all strains as released?
Yeah, so before I left, we sent off our samples for whole genome sequencing. So hopefully by the time we come back, maybe that matter will be the same.
Great.